Hey everyone, so this is part two of our notes, and this focuses on the generation of a nerve impulse. So this is actually the process by which an electrical impulse is created within your neurons. All right, so if we look at this picture over here, you've got sort of two different neurons. The first neuron is myelinated, right? So this one, myelinated. I'm spelling this wrong, so I'll fix that. So this myelinated neuron, you can see has those Schwann cells, right? Remember, those are those fatty cells that are wrapped around, and you can see how it's wrapped around here in like kind of that crepe-like fashion, like I mentioned before. And these myelinated neurons are found linking the peripheral nervous system to the central nervous system. And what's happening here is the electrical impulse is traveling very long distances. And so by myelinating this and creating, remember, those nodes of Ranvier, what happens is we're going to see the action potential only occurs at these nodes of Ranvier. So that causes the electrical impulse to jump from one node to the other. And this is what we call saltatory conduction. Saltatory conduction, which is literally the jumping of the electrical impulse from one node to the next, and this helps speed up the signal, right? And that occurs in these myelinated neurons connecting the peripheral nervous system to the central nervous system. Down below in this other one, we have an unmyelinated, right? So we can see that there's no Schwann cells, it's unmyelinated, and this generally occurs within the central nervous system from one neuron to the next and traveling uh, uh, not great distances, right? So this one is long distance, right? And this one is very short distance, okay? And what we're going to start with here is looking down here. We're going to look at this graph and just kind of go through the names of the processes. And then on the left-hand side, you can see over here, we're going to talk about each individual set of steps in this, what we call action potential. So starting with step one, which maybe isn't really a step at all so much as kind of a maintenance, and that's what we call resting potential, okay? And this is an action taken on by neurons that helps maintain what we call the membrane potential. So you can see over on this side on the y-axis we have membrane potential, and this is time in milliseconds of which an action potential is generated. So we have resting potential, which is the first part of the action potential, or not even so much the action potential, but just the status of the neuron when it's not stimulated. So then we have at this point some sort of stimulation has occurred and it's causing the membrane potential to increase. So remember over here, we're looking at about a resting potential of around minus 65 to minus 70 millivolts, right? And so some sort of stimulus causes the action, or sorry, the membrane potential to increase. And when it reaches this threshold, which we'll talk about, somewhere around minus 55 millivolts, that threshold sort of surpasses the threshold and then causes depolarization, which is what we'll call step two in this overall process. And so the membrane potential is changing from this minus 65 and it's going up to about plus 40 millivolts. And we'll talk about what's going on there, but then it enters into the third stage of the action potential, and that is what is called repolarization. And so now the membrane potential drops back down, and it actually tends to overshoot the mark, heading upwards of minus 70 millivolts. And then we enter in, that's referred to as hyperpolarization, right? So think about those words, right? Remember, polar, opposite charge, right? Opposite sides. So the membrane has opposite charges distributed across the membrane. So depolarizing sends it from negative to positive. Repolarizing sends it from positive back down to negative. And this hyperpolarization, if we think of hypertonic, right? It's like excess polarization. So it sort of overshoots its mark. And this is what we call the refractory period. And then we get back to step one, the resting potential. Okay, so that's sort of the distribution of the stages of an action potential. So starting with number one here at the top, so we're going to break each one down. So we've got resting potential, and I'll just slide that down, make sure you can see all that. I'll erase this part here. So resting potential, this goes way back to when we talked about membrane transport, and we learned about the sodium-potassium pump. So that's this guy shown in blue right here, sodium-potassium pump. So resting potential is a membrane potential of around 65 millivolts. And I'll put that, sorry, this is my approximate symbol, so I'll put approximately negative 65 millivolts. It is maintained by the sodium-potassium pump, maintained by sodium-potassium pump. Okay, so this sodium-potassium pump is pumping so it pumps 3 sodium ions out and 2, if you remember, 2 potassium ions in. Now, what that does is it maintains this. So although the primary function of the sodium-potassium pump is to move those ions, so sodium ions are going out, right? We got sodium ions going out, and we got potassium ions going in. There is other ions, and I'll just show this image here. So if you look over here, we can see that there's other ions that contribute to this overall charge. So over here we got this little voltmeter. So we got one side of the voltmeter connected outside, extracellular, and then we have the other side connected within the plasma membrane here, and across the cytosol, which is the inside. And so what we see is we're going to have some positive charges here due to the potassium ions, but you see there's a huge negative charge inside, and that's due to a number of proteins that carry some negative charge, as well as other ions inside the cytoplasm of the neuron that have a negative charge. Outside, we see also that there's some negative, but predominantly a positive charge on the outside. So what this does is it gives us a resting membrane potential of -70. So the inside is negative 70 millivolts relative to the outside. All right. So that's resting potential. It's maintained by the sodium-potassium pump. And so any neuron that has not been stimulated is sort of sitting at this resting potential. So then a stimulus occurs. And when a stimulus occurs, you get what we call depolarization. All right. And depolarization is characterized by, so a stimulus causes sodium-gated channels, sodium-gated ion channels. Now this should ring a bell back to, again, membrane transport. This is facilitated diffusion. There's no energy involved here. And maybe I didn't emphasize that or remind you that this sodium-potassium pump is active transport, right? So ATP required, right? And that's because, again, we're moving against the concentration gradient, right? Against concentration gradient, moving sodium ions out, potassium ions in, and you're pumping them against the gradient to try and create a high concentration on either side. But in the case of the depolarization, so the beginning of the action potential, what we've got is a stimulus causes these sodium gated channels to open. We get an influx of sodium ions, right, traveling down the concentration gradient. So they're traveling in. This positive ion charge traveling into the membrane is causing the membrane potential to go from that minus, so the membrane potential goes from minus, 70, let's say minus 65. So minus 65 millivolts to plus 40 millivolts, right? As the influx of sodium ions goes into the neuron. And that's depolarization. Sometimes you might see it referred to as the upswing in the action potential in some books, but definitely make sure you understand depolarizing, all right? With the cause of, or with the result of depolarization, this then leads to step 3 of the action potential, which is referred to as repolarization. All right. And so repolarization is characterized by the opening of potassium gated channels. So potassium gated channels open, and there is potassium ions move out of the neuron. Again, it's just facilitated transport, so there's no energy involved, just the movement from high to low concentration. As those sodium ions, or sorry, as those potassium ions move out, we are now, the membrane potential is going back down, all right? And oftentimes what happens is it surpasses the resting potential. So you get this slight decrease below, right? And we get the plus 40 millivolts is now going to, you know, approximately minus 70 millivolts. So just kind of shooting past that resting potential of minus 65 millivolts. And this is referred to as, at this point where it's slightly past, we refer to this as hyperpolarization, right? Hyperpolarize. Sorry, I'm writing over this. Hyperpolarization. Now, what's happened here is the membrane potential is being brought back, but we enter into this step 4, which is this refractory period in which there's no possibility for the membrane or the neuron to generate another action potential. All right, so this is the refractory period. And although the membrane potential is back down to where it should be, the neuron can't fire again because the ions are on the wrong side. What you've got here is you've got all the potassium ions on the outside, and you have all of the sodium ions on the inside. So remember, sodium ions have come in, potassium ions have gone out, and now we're sitting right here. But the membrane cannot fire again because the ions are on the wrong side. So membrane or neuron is unable to generate an action potential. All right, so membrane or neuron is unable to generate an action potential. Sodium and potassium ions are on the wrong side. And in fact, from this point onward, really from this peak in the action potential until it's back to the resting potential, this is all referred to as the refractory period, in which case no new action potential is able to be generated once a neuron is stimulated beyond the threshold, which I'll talk about in a second. Once it's stimulated beyond the threshold, all the way through here, another action potential cannot be generated. And so this refractory period is kind of divided into two stages, but ultimately we just need to understand that this time period here from about 3 milliseconds on to about 5 or 6 milliseconds, no action potential can be generated again. I guess it's about maybe 2.5, right? 2.5 milliseconds onward. So then finally we're back to the resting potential, and we can just say see above, right? Where now the sodium potassium pump is going to kick in, and it's going to be pumping in those potassium ions back in and the sodium ions back out to their correct positions and restoring that membrane potential back to the minus 65 millivolts with the ions on the right side. Okay, now a couple little things here. First off, this threshold. So let me just erase some of this stuff here, clean this up. But what you see here is an action potential is an all or none response. And essentially, the stimulus has to cause, what you need to remember is a neuron doesn't just have one sodium gated channel, right? It has many. And different stimuli may only cause some sodium gates to open, in which case, if a minimum amount of potential difference is not achieved, no electrical signal, no action potential will be generated at all. So a very, very small stimulus may not lead to an action potential. And we'll see that it's actually a cumulative effect of stimuli that eventually, once a threshold is achieved, and the threshold is generally around minus 55 millivolts, once that stimulus causes a membrane potential of minus 55 millivolts, then that just triggers sodium gates opening, like opening up the floodgates, and then action potential is generated. So we call this an all or none, right? Minimum threshold in order to generate minimum threshold minus 55 millivolts required to generate an action potential. Now, the last thing I'll leave you with, and we'll review some of this tomorrow, or in the next class, but every action potential is the same. It doesn't matter whether the stimulus was more intense, like, I put my hand on an extremely hot stove versus, light entering into our eye or sound waves entering into our ear, or the change in your pH of your blood or change in CO2 levels. It doesn't matter what the stimulus is, the action potential is the same. What differentiates the intensity of the stimulus is the frequency. Okay, so something that is much more intense, like say hand on the hot stove, will generate action potential after action potential after action potential very rapidly, which relates to the intensity of the interpretation or the integration of the sensory input, right? Okay, that's it for action potentials, and make sure you bring any of your questions.